This is the study we did a couple years ago. It's because FLT3 ITD is one of the most common mutations in AML, acute myeloid leukemia. Then it's a mutation in FLT3 ITD is a very common mutation. Then a lot of groups actually developed a specific inhibitor, FLT3 inhibitor, targeted FLT3 ITD, then effectively inhibit the activity of FLT3, then basically killing the AML cells. But clinically, there's actually many FLT3 inhibitors in clinical trial. There are at least 2 already approved by FDA as a clinical drug to treat as monotherapy, even treat the AML. But clinically, during the clinical practice or even in clinical trial, there's a drug resistance phenotype observed. So obviously that's happened to every drug to AML. So then for our study, the purpose is to understand the drug resistance, molecular mechanism underlying this drug resistance. So we did the whole genome CRISPR-based screening. CRISPR technology obviously is a state-of-the-art technology, so very effective to knock out genes, whole genome. Then we identify a bunch of genes. On top of the list, there's a SPRD3, SPRD, one of the SPRD member, and also GSK3, glycogen synthesis kinase 3. We even identified also signaling pathway regulated by SPRD3 and GSK3, namely RAS, MAP kinase signaling, and the Wnt signaling. So then we also confirmed this in clinical samples, AML samples, to show that actually those genes downregulated in the FLT3 inhibitor resistant samples. Then also in the paired samples, which before the treatment, then after treatment develop resistance, they have downregulation of the signaling pathway and also downregulation expression of genes. So this obviously pointed to the probably new therapeutic approach to treat the AML, especially FLT3 inhibitor resistant AML. So we can add on probably MAP kinase inhibitor or beta-catenin inhibitor with targeted Wnt signaling, then maybe use a combinational therapy then to better treat the FLT3 ITD positive AML. Yeah, that's our future goal. Yeah.